Hello, everyone. My name is David Vaillancourt. I'm a professor at the University of Florida and also Chief Scientific Officer at Neuropacs. And today I wanted to talk to you a little bit about some of the software that we've developed, which is out of research ongoing for a little over a decade now. And it's really in both the Parkinsonism and dementia space, but I'll spend most of the time today talking about Parkinson's disease and Parkinsonisms and then touch upon the dementia part at the end. So, as many of you know already, the standard of care for diagnosing Parkinson's disease and different forms of Parkinsonism is patient sees doctor. Doctor may or may not order an MRI. In our market research, most of the patients do get an MRI already, and it's typically to rule out a tumor, stroke, or vascular Parkinsonism. They're not typically used. MRIs are not typically used proactively to determine diagnostics. And so currently, diagnostic accuracy in early stages of PD, if you look at the literature compared to neuropathological confirmation, is only 55 to 75% accurate. It does raise to up to 85% accuracy after 5 years. And so part of what we're trying to do in our software is typically that MRI, after it occurs clinically and it rules something out, it's thrown away or it's never used again. And part of what we're trying to do is start to use it proactively. We believe with AI and certain types of imaging modalities that one can use it proactively to help the doctor make the more accurate diagnosis. So diffusion MRI is the technique that we're focused on. This particular technique is standard on just about every 3T scanner worldwide today. Siemens, Philips, and GE scanners have routine FDA-approved sequences to run diffusion MRI. And so it's really looking at the way water molecules diffuse in the tissue or in fluid. And so in a fluid environment, there's going to be unconstrained diffusion of water molecules. And then in a constrained environment, like you might imagine, like white matter in the brain, it's going to diffuse along a specific track or a specific orientation. And the constraints on that diffusion process are very relevant because they change in different neurodegenerative diseases and different conditions, certainly relevant to Parkinson's disease and forms of Parkinsonism. So we've been using a technique which is called free water imaging. And in free water imaging, it is effectively looking at the way water molecules are diffusing and assuming two different sort of compartments in a voxel. And so in blue, it shows the extracellular compartment, and that's the free water compartment. And the measure is the fractional volume of free water within that compartment. So you can imagine that as that compartment grows, the more of that voxel is taken up by unconstrained diffusion, which is really fluid. The circles represent cells, and so that's the tissue compartment. And there's an assumption that there is in a tissue compartment. And then we are effectively using other measures from diffusion tensor imaging, particularly fractional isotropy, which is looking at the degree of direction-dependent diffusion. And so we are using those two measures, free water and also free water corrected FA, to kind of better understand neurodegeneration going on within voxels, which we think are highly relevant for brain regions and also highly relevant for disease state and also progression. So I use this as an analogy just to draw your attention to why free water imaging is more sensitive than volumetric MRI. So, if you think of a T1 weighted MRI, you would measure volumes of the brain. And this particular method is actually used quite frequently in the Alzheimer's disease field because you can measure hippocampal atrophy, of course. Whereas in Parkinson's disease, the atrophy is not typically occurring very early on. It might occur later on in the disease process. But diffusion MRI is picking up on something that's more sensitive than volumetric imaging. So volumetric MRI, might look at this outside of this building and notice that the outside of the building, while decaying a little bit, it's not decaying that much. The windows on this building still look pretty good. But if you look at the bricks throughout the building, you start to see decay or degeneration going on in many different parts of the building. And diffusion MRI is looking at all the bricks and all the degeneration going on, whereas a volume is only looking at the outside. And so diffusion MRI by nature is going to be more sensitive than a T1 weighted volume type metric. And so we've shown this in the substantia nigra. This was a paper in 2015 looking at free water in the substantia nigra. You see the blue here in this individual patient shows increased free water bilaterally in the substantia nigra. And this particular image just to the right is the same PD patient 1 year later. And so you can appreciate that in the control group, while there's no change over 1 year period of time in free water in the posterior part of the substantia nigra, the the PD patients do have an increase over that 1 year period of time. And so at that time, this was the first study that ever showed progression of substantia nigra changes in humans longitudinally. So since that time, we published a paper in 2017, which accessed the data from the Michael J. Fox Foundation, the PPMI study, where effectively we asked the question: Can we replicate what we saw in our cohort, which is the University of Florida cohort? Do we also see that in the Michael J. Fox Foundation cohort? And so the y-axis here in part A is showing the 1 year change in the de novo PD patients over a 1 year period of time compared to the control group. And you can see that the yellow shows the control group where there's no change over that 1 year period of time, whereas in PD, there's a significant increase in posterior substantia nigra free water over a 1 year period of time. In addition, in a separate cohort, which is studied longitudinally over a 4 year period of time, this is PD only, you can start to see sort of progression changes in the substantia nigra over a period of time. So there's a significant increase from baseline over 1 year, 2 year, and also 4 years progressively in these PD patients in the PPMI cohort. It's also important to note that this is an international study across many sites. So in either of these, so there's no group by site interaction in A, and there's no time by site interaction in B. And that starts to suggest that these measurements and these metrics can start to be utilized at multi-sites. And that's relevant because any phase 3 clinical trial has to be multi-site. So for any of these techniques that I'm showing you to be relevant in clinical drug development or in pharma type trials, multi-site is really quite important. So this is a study from a group in China where they looked at free water in the posterior part of the substantia nigra in RBD patients and RIM behavior disorder patients. And you can see in the y-axis here in A is the posterior part of the substantia nigra, where they show an increase both in RBD patients relative to controls and also PD patients relative to controls. Whereas if you look at the anterior part of the substantia nigra, there's not a significant change in either of any of these groups. We've shown in other evidence, I'm not going to go into that in detail today, but the anterior part of the substantia nigra becomes affected later on in the disease process of PD. So early on, we see these posterior part of the nigra effects, whereas the anterior part of the nigra starts to become affected later on in the disease process. And then in another study, which has just come out last year, it's a very interesting study where they're following a group of RBD patients and then looking at conversion and phenotypic conversion in the RBD patients over many years. So you can see the follow-up time in A and B. These are follow-up times for some patients between, just 4 years, 8 years, 12 years, and up to 16 years. So these are long periods of time where you're starting to look at conversion. And you can see that the baseline imaging for the nucleus basalis and minor and also the posterior part of the substantia nigra for free water, those with higher free water content had a more likely probability of converting to either DLB or PD when looking at nucleus basalis and minor and posterior part of the substantia nigra. So what this is teaching us, I think, is that these free water signals are changing really early on in the disease process. So the fact that you're picking these up at a baseline scan in an RBD patient and then they're predicting phenoconversion years later strongly suggests that the free water signals, certainly in the nigra, and also nucleus basalis and minor, are becoming very important predictors over time. So in 2019, we published a paper in Lancet Digital Health, which was like a real-life messy data set. And in this data set, we put together data from 17 different MRI scanners, 8 different international studies. PPMI from Michael J. Fox was one of them, but there's many others on here as well. And we asked the question, can we differentiate Parkinson's disease from multiple system atrophy and progressive supranuclear palsy? We studied MSA and PSP in comparison to PD because MSA and PSP are the most frequently misdiagnosed neurodegenerative diseases compared to misdiagnosis for PD. And so we put all this data together and looked at a machine learning algorithm and just asked the question, can we use a machine learning algorithm, which is just based upon diffusion imaging, to differentiate these types of diseases? And so part A and at the top here on the upper left is showing PD versus atypical forms of Parkinsonism. And I'm going to go to part B here, which is basically the training and validation AUC. So you can see strong training and validation, diffusion imaging plus UPDRS, diffusion imaging alone, and also UPDRS alone. And if you look at the test AUC in the bottom here, again, you see high AUCs in the diffusion imaging data comparisons compared to just the UPDRS only. Now, that's not to say the UPDRS is used for diagnosis. That's not the point. And we use the UPDRS as an add-on here because we wanted to see if the severity of the patients was driving some of the signals we're seeing. And since you see in the middle, the diffusion only has a similar AUC as diffusion imaging plus the UPDRS, it doesn't seem the UPDRS is adding much to the ability for diffusion imaging to differentiate these groups. Now, we also looked at a different model, which is MSA versus PSP. And in F and H, you can start to see that, again, diffusion imaging alone is distinguishing MSA and PSP with high levels of accuracy. So what this taught us in this study was that this is possible. I think it kind of, sometimes in research, you start to realize that when you're doing something, it actually might have clinical applicability because this is a data set where the scanning sequences are not matched, the head coils are not matched, the scanner vendors are not matched. It's a pretty real life messy data set. And so it starts to teach us that in principle, this is feasible to look at this and detect clinically relevant differences between these disease groups that could be relevant going forward. And so what we did in this next study was we wanted to test that prospectively. So we designed a study in concert with the Parkinson's study group, where they advised us on the study design, and also the study was funded by the NIH. And so this is a study where we looked across 21 sites. We had 315 prospectively tested patients. The map of the sites is shown here on the right. 19 sites from the US and 2 were in Canada. We had 3 blinded neurologists, movement disorders trained neurologists, provide the gold standard. So the referring neurologist, for example, might come from UC San Diego, and then they would come into a database, and Dr. Oken from University of Florida and also Dr. Tauzi from University of Chicago would blindly look at those videos, MRIs, but also the clinical features of these patients to also give their own diagnosis. Now, none of them had access to the AIDP algorithm. They only had access to the clinical information in a standard MRI that you would collect clinically. In this study, we also had about 11 Siemens scanners, 5 GE and 5 Philips in the study. And then we had another cohort, which was 49 brains, where we had imaging prior to pathology or prior to patient passing. So we had autopsy confirmation. And so we wanted to test the algorithms against that pathologically confirmed cohort as well. This shows the consort diagram. This paper was published in JAMA Neurology in March of 2025. And so you can see 316 patients were screened into the study. We excluded some, and these were exclusions because the agreement amongst diagnosis was not agreed by the physicians amongst all 3 physicians. And based upon the study design recommendations from the Parkinson's study group, we needed to have an established reference standard where all 3 physicians would have an agreed upon diagnosis. So then we randomized 249 into the study. Some went into the training set, and some of the training set also came from a retrospective cohort as well. So we had 500 randomized to the training set, and then we had another 145 into the testing set. Now, the 145 that are in testing set did not go into the training set ever, so they were completely separate, and there's no bleeding between training set and the testing set. So this is the primary outcome data, which is showing you the first model, which is PD versus atypical. The area under the curve was 0.96. MSA versus PSP, area under the curve was 0.98. PD versus MSA model, 0.98, and then PD versus PSP was 0.98. So these are all separate machine learning models. They all have the same parts of the brain in each model, but the weightings that are doing the classification are going to have different weights. And it's effectively, you could think of it as like a fingerprint detection. We're basically detecting the degenerative parts of the brain that are indicative of an atypical form of Parkinsonism. Or we're in the MSA versus PSP, we're detecting the PSP types of degeneration that are different from MSA types of degeneration. So what we wanted to do here was we thought possibly that the test cohort that we had in the previous slide might have just been lucky, right? So it's possible that the test cohort was just by definition, not representative cohort. So we wanted to run the experiment again and again and again. So what we decided to do was, like in a post hoc way, we wanted to changed the test cohort so that we could then determine if we got similar outcomes. And so all of these lines represent 49 new runs, and the average AUC is shown here with the range. So PD versus atypical had about 0.94 on average. MSA versus PSP was again 0.98. PD versus MSA is 0.94, and PD versus PSP is 0.96. So by and large, when we reran the experiment many times, we came up with a very similar result in terms of efficacy levels for the different model runs. And then finally, when you think of looking at this in the context of the autopsy validation cohort, so we were able to look at 49 autopsy confirmed cases, and the AIDP algorithm predicted 46 of 49. So it was about 93.9% accurate. And that was over a 12% diagnostic gain compared to the final clinical diagnosis. So even in these autopsy confirmed cases where physicians have followed these patients for many years, these are years and years that these physicians have followed these cases, they still were only 81% accurate in final clinical diagnosis, matching up with the pathological diagnosis. So this is a study that's under review, kind of interesting data set where we looked at the PPMI cohort. You can see the AIDP classification on top, and we wanted to compare this with the CSF assay looking at synuclein positivity or negativity. So this is the full cohort that we had access to, 491 individuals. And you can see in that cohort, which is the PPMI cohort, 92% were identified as PD by our algorithm, whereas 8% were identified as a typical form of Parkinsonism. If you look at the synuclein positive group, you see similar percentages. So those that are positive with synuclein had 93% identified as PD by the AIDP algorithm, whereas 7% was atypical. And for those that were SA negative, 88% were identified as PD by the algorithm, whereas 12% were identified as atypical. And then similarly, when you go down to the bottom, and this is looking at synuclein status, so synuclein positivity in that cohort is about 89%, whereas 11% are synuclein negative. Whereas in the AIDP PD-only cohort, they're synuclein positive at 90% and then 10% negativity. And then the AIDP, where the atypical form was identified, 84% are synuclein positive, whereas 16% are synuclein negative. So the suggestion here is not that the AIDP is to necessarily competing with looking at synuclein positivity or negativity. We view this as a sort of a concert, basically to be used with each other. So the idea is that the AIDP is picking up neurodegenerative patterns, and these neurodegenerative patterns are very useful, possibly when compared with a synuclein type marker. And we view these as both useful in comparison to each other and not necessarily in competition with one another. So I just want to quickly switch and tell you a little bit about dementia very quickly in a recent paper that we published. So we've shown that free water is more sensitive than volume in a publication in 2019. I'm not going to go into the details, but it's more sensitive in the early stages of detecting changes in early MCI compared to controls when comparing free water in the hippocampus compared to volume in the hippocampus. And so we wanted to apply the same principles that we did in the AIDP to a dementia cohort. We looked at 730 diffusion images. We excluded some. The key one I want to talk about here is that we looked at amyloid. No amyloid negative AD patients were in here, so they all had to be amyloid positive. And we also excluded amyloid positive DLB in this cohort because we wanted to first focus on amyloid negative DLB cases compared to amyloid positive AD cases. So we had 519 total images. We had 258 Alzheimer's disease, 129 DLB, and 132 controls. This shows the AUCs for training and validation for AD versus DLB. That's the first one. You can see the testing AUCs are very high. AD versus control, again, testing AUCs are high. DLB versus control, again, testing AUCs are a little bit lower, but they're still well over 0.9. And then control versus AD DLB, again, testing AUC is a little bit lower, but it's still well over 0.9. And then finally, I wanted to sort of close and show you the comparison of pathology. So in this case, we only have 13 cases in this study, but I wanted to show you the results from that. So the final clinical diagnosis of the pathological cohort here was only 54% accurate. And you can see the different clinical diagnoses here. They range from dementia with Lewy bodies to Parkinson's disease to cortical basal syndrome to Alzheimer's disease. The pathological diagnosis shows Lewy body disease or Alzheimer's disease in these 13 cases. And you can see the AIDD, which has AD versus DLB probability, the differential diagnosis probability algorithm is 100% accurate in each one of these. So it seems as if the AIDD algorithm is having high levels of accuracy. And now whether it would be sustained 100% is certainly unlikely. But in this early stage small cohort, it seems to be quite promising in predicting pathological accuracy. So today I've shown you a number of things. And just to summarize, free water is sensitive to early changes in the nigra and in the hippocampus. The AIDP is detecting atypical forms of Parkinsonism in a multi-site cohort. The AIDP works across Siemens, GE, and Philips 3T scanners. The AIDP is also in agreement with a neuropathology cohort. And then finally, the AIDD, which is in the dementia, is highly accurate in differentiating DLB from AD. I just wanted to say thank you very much for your time and your attention. This is our emails. If anybody's interested, we're working in the clinical trial space, but also and moving this software into clinical space as well as the goal. But we're very interested in collaborating and working with folks on the call.